Hi, today I'll talk about the prediction of bucc-snoRNA interactions in two organisms. Bucc-snoRNAs, or small nucleolar RNA, are small non-coding RNAs well conserved in eukaryotes. They are characterized by the presence of well-conserved pattern called the boxes C in C' and D in D'. They interact with specific proteins for stability and function. Their canonical partners are NOP56, NOP58, SNU13, and the methyltransferase fibrillase. But some snoRNAs have been shown to interact with splicing factors such as RBFOX2, polyadenylation factors such as FIP1, and an acetylase, CRE33, and other proteins. Their canonical function is the methylation of ribosomal and small nuclear RNA. But in recent years, novel diverse functions were shown for bucc-snoRNAs, such as the regulation of alternative splicing by RNA competition, the regulation of 3' processing by the interaction with polyadenylation factors, regulation of the RNA level, and RNA acetylation. Currently, there are 140 validated canonical interactions and only 16 validated non-canonical interactions. Canonical interactions are mostly with ribosomal RNA, whereas non-canonical interactions are mostly with protein coding genes. The study of non-canonical interactions is difficult due to their vast diversity and the lack of appropriate tools. We reanalyzed the dataset of high-throughput RNA-RNA interaction identification methodologies called Paris-Liger seq and SPLASH. These methodologies allow the detection of all RNA-RNA interactions in the cell. To do so, they use a reversible psoralen cross-linking. Then they purify the double-stranded region. They do a proximity ligation, then reverse the cross-link and sequence the RNA duplex. Doing so, we were able to identify almost 500 non-redundant bucc-snoRNA interactions with a wide range of RNA targets, showing that the non-canonical interactions are more widespread than we would think. We then used all these interactions, so the interactions from the high-throughput RNA-RNA interaction methodologies, the non-canonical and the non-canonical interactions to build snoGlobe, which is a bucc-snoRNA-RNA interaction predictor. So snoGlobe is based on a gradient boosting classifier, which is a combination of multiple weak decision trees. To build snoGlobe, we split every interaction in 13 nucleotide sliding windows, which means that we took the first 13 nucleotides of the interaction, then moved one nucleotide, took another 13 nucleotide window, then moved one nucleotide, took another 13 nucleotide window, and so on until the end of the interaction. Then, for every positive window, we created 20 negative examples, 10 of which were completely random RNA pairs, and 10 were matched negative examples. The matched negative examples originate from the same gene pair matching a positive window. For each positive example, one matched negative has the same position in the snoRNA and a different position in the target. One has the same position as the positive window in the target, but a different position in the snoRNA. And 8 matched negative examples have random positions, but in the same gene pair as the positive window. The positive, matched negative, and random negative windows were split in 3 sets for hyperparameter tuning, training, and testing. For each example, specific features were extracted, such as the relative position in the snoRNA, the sequence of the interaction, the target biotype, and the position in the target. These were used as input feature for snoGlobe. Using the test set we set aside, we compared snoGlobe to Plexi and other bucc-snoRNA interaction predictor and to generic RNA-RNA interaction predictors, which are RNA-Plex, IntRNA, RiseRh2, and RNA-Up. As you can see in the ROC and precision and recall curves, snoGlobe in blue performs better than the other tools on the test set. To further compare the tools on a similar basis, we use thresholds to obtain 90% precision with each tool. Comparing each tool with these thresholds, we can see that snoGlobe retrieves the higher number of snoRNA interactions, but also the higher number of each category, which are the high-throughput interactions, the validated canonical interactions, and the validated non-canonical interactions. We can also see that snoGlobe is the only one to retrieve most of the high-throughput RNA-RNA interactions and the validated non-canonical interactions. So snoGlobe works great in human, but since its development, the snoRNA community has shown great interest in using it with different organisms. So to assess its potential in yeast, we compared the result of snoGlobe's prediction on high-throughput RNA-RNA interactions retrieved by CLASH. So CLASH retrieves RNA duplex through the UV cross-linking of specific proteins. In the case of boxy snoRNA, they used canonical boxy snoRNA interactors. The interaction retrieved by CLASH are mostly ribosomal RNA, protein-coding genes, and snoRNA. So we used snoGlobe with a probability threshold of 75%, and we were able to retrieve almost half of CLASH-identified interactions from most target biotypes. Looking more closely at snoGlobe's prediction for CLASH interactions, we can see that longer interactions tend to have a better score given by snoGlobe. We can also see that snoGlobe gives a high score to interactions with ribosomal RNA, but also with protein-coding genes and other snoRNA, which is great since our main goal is to retrieve all kinds of snoRNA interactions. This data is still preliminary, but we will continue to investigate snoGlobe's performance in yeast. In conclusion, non-canonical interactions represent an important part of boxy snoRNA interactions. snoGlobe performs very well in human and is able to retrieve a higher number of interactions of every category than all the other tools tested. In yeast, snoGlobe is able to retrieve a good proportion of CLASH-identified interactions from most biotypes. Thank you for listening.